Welcome to Group 10's e-tutorial on the regulation and the function of the electron transport chain. So the electron transport chain is the endpoint of aerobic respiration that's found in mitochondria, and it also has a part in photosynthesis in chloroplasts and also in cyanobacteria and purple anaerobic bacteria. In mitochondria, it occurs primarily on the inner mitochondrial membrane, and in chloroplasts, it occurs on the thylakoid membrane. Then in bacteria, which don't have membrane-bound organelles because they're prokaryote, it occurs on thylakoid-like structures, which are folds in the membrane, or folds in the outer membrane, I should say. So now we have a short video to show you that is an overview of the electron transport chain in mitochondria. Unfortunately, it's not quite as simple as we've just seen, so we're going to go through it on a complex by complex basis. The first complex is NADH coenzyme Q oxidoreductase. This is what it looks like in real life. You can see it's got a large hydrophilic domain and this transmembrane hydrophobic domain, and that's the one that's important during proton translocation across the membrane. There's an NADH, which has been produced by the citric acid cycle, and that is oxidized to NAD by a flavin mononucleotide in the hydrophilic domain. This causes release of 2 electrons in the form of hydride ions, which pass down to a Resk ion protein, which contains 9 clusters. However, as you can see, only 7 of these clusters actually participate in the transport of electrons. The other 2 kind of indirectly affect it by affecting redox potentials. So there's your 7 ion clusters right there. And now there's a ubiquinone that's in the membrane, and that is reduced to ubiquinol, which can then diffuse through the membrane to complex 3. This process causes a change in the conformation of the hydrophilic domain, and this change in conformation causes a hydrogen ion to be directly transported at the interface between the hydrophilic and hydrophobic domains, and it also causes a change in conformation in this sort of horizontal transmembrane arm that spans through the hydrophobic domain, and that causes an alignment of 3 proton transporters in that domain that then subsequently causes the transfer of the remaining 3 protons per cycle that complex 1 transports. So here's just a quick summary of complex 1. There'll be one of these for all of the complexes. Now we'll move on. So in the TCA cycle, there is a conversion of succinate to fumarate, which converts FAD into FADH2. The FAD is actually covalently bound into complex 2, which looks like this. And when it oxidizes succinate to fumarate, it is then itself oxidized back to FAD plus by donating its electrons to a series of iron sulfur complexes, 3 of them to be specific, and then from here the electrons are transferred to a ubiquinone, which is reduced to ubiquinol. Complex 2 also contains a bound heme B556 group, and when electrons are actually passed down through the iron sulfur clusters, they're passed through one by one, and this leads to the formation of a semi-quinone intermediate, which is highly reactive and can form reactive oxygen species. And to prevent this, the electron is sort of quickly transferred between the heme group and the semi-quinone intermediate to prevent the chance and reduce the risk of forming reactive oxygen species. It's worth noting that complex 2 is the only complex that doesn't transfer protons across the membrane, so it doesn't contribute to the proton gradient, it just provides a pathway for electrons to get into the quinone pool. The ubiquinol, formed from the last 2 complexes, moves through the membrane to complex 3, where it is oxidized to ubiquinone. Thus, complex 3 is called coenzyme Q cytochrome C oxidoreductase. It's the site of a process called the Q cycle, and during the first cycle, a quinol binds at the QO site and is oxidized and releases 2 protons which are transferred into the intermembrane space. The 2 electrons released from this oxidation are transferred by 2 separate pathways. One is transferred to an iron sulfur protein and is then transferred to cytochrome C1. Then from cytochrome C1 it is transferred to the mobile carrier cytochrome C. The second electron is instead transferred to cytochrome B, to the Bl heme, and then to the BH heme, and from there it's transferred to the QI site where a quinone molecule is bound. The quinone accepts this electron and is reduced to a semi-quinone intermediate. A second quinol will then bind at the QO site and the cycle is repeated. 2 more protons are released and a second cytochrome C is reduced, and at the end of it the semi-quinone intermediate at the QI site is reduced to quinol. This process happens like this because ubiquinone can transport 2 electrons, but the mobile carrier cytochrome C can only carry 1 electron, so there needs to be some kind of process that allows the conversion from a 2 electron carrier to a 1 electron carrier. Complex 3 is also thought to be the major site of reactive oxygen species production in the electron transport chain. So the next step in the chain is complex 4, which oxidizes cytochrome C and eventually transfers the electrons to molecular oxygen to form water. Because cytochrome C can only transport 1 electron per molecule, it takes a total of 4 cytochrome C molecules to convert 1 oxygen to water. This is what complex 4 looks like. And first of all, then you get a oxidation of cytochrome C to transfer the electrons to copper A. This then transfers the electrons to a heme group. The next stage in complex 4 is actually a binucleate complex formed of heme A3 and copper B. The electron is transferred to this binucleate complex, and from there an oxygen molecule binds. 4 protons then react with this oxygen to form 2 molecules of water, which are subsequently released into the matrix. The exact mechanism of this is as follows. It's not necessarily needed, but if you're interested you can follow that link there to an article that tells you all about it. Starts up here with part A. So Oxygen is reduced to water by a series of redox reactions that occur in complex 4. However, the energy from these reactions is also used to translocate 4 protons from the matrix to the cytosolic side of the mitochondrial membrane. So how does the complex sort protons that are going to the active chemical site from protons that are going across the membrane? And also, how does it stop protons that have already passed across the membrane just travelling back through the complex? Well, it's got 2 channels within the complex, the D channel and the K channel. The K channel transports 2 of the chemical protons to the reactive site. The D channel transports the other 2 to the active site. However, only the D channel can transport protons to the cytosolic side of the mitochondrial membrane because it's the only channel that aligns with the exit. The D channel begins with a aspartate residue at position 91, and the K channel has a lysine residue at position 319, and that's where they get their names from. The D channel also contains a glutamate residue at position 242 that is key to its activity. The glutamate residue in the D channel and the lysine residue in the K channel point into the channels themselves and are aligned so that they point downwards towards the entrance to the channel. And when protons bind to these residues, there's a change in conformation that causes them to flip up towards the centre of the protein and the active and the pump site. When they are deprotonated, they then flip back down to their original position, which means that protons which have gone past them and are in the active site can no longer rebind to them to travel back through the entrance channel because the conformation changed and the residue is now too far away for the reaction to occur. The protons are sorted between the active and the pump sites using a chain of water molecules, and the orientation of this chain of water molecules, either to the pump or to the active site, depends on the reduction state of different proteins within the complex. So if the heme A3 copper B binucleic complex is reduced, then the water chain will be orientated to transfer electrons to the active site. But if the heme A is reduced, then the chain will be orientated to deliver electrons to the pump site. It's also worth noting that the lysine residue at position 319 is also thought to have a role in breaking of the dioxygen bond during reduction of oxygen to form water. The final complex, which isn't technically part of the electron transport chain, but is worth mentioning because of the effect it can have on the electron transport chain, is ATP synthase. ATP synthase is made up of 2 domains, the F1 domain and the F0 domain. F0 is insoluble and spans the inner membrane of the mitochondria. It's made up of the A subunit and a variable number of C subunits that are formed into like a ring shape. The peripheral stalk subunits, B, F6, D, and the oligomycin sensitivity conferral protein, OSCP, are also present in the F0 domain, as well as a number of other accessory subunits, including subunits E, F, G, and A6L. These subunits are important because ATP synthase actually normally exists as a dimer or as an oligomer when on the inner mitochondrial membrane, and it's these subunits that allow interaction between the ATP synthase molecules to form these like super complexes. The F1 subunit contains 3 alpha and 3 beta subunits which form an alternating ring structure, as well as a single gamma, delta and epsilon subunit. As you can see, the gamma subunit protrudes up into the alpha beta ring and is anchored to the transmembrane C ring by the delta and epsilon subunits. The alpha beta ring is fixed to the beta subunit, which is then in turn fixed to the A subunit. In ATP synthase, it is actually the beta subunits that catalyse the formation of ATP. They cycle between open, tight and loose configuration, and it's the transition between these states that actually provides the activation energy for reacting phosphate with ADP. The energy required for the conformational changes of the beta subunit is provided by diffusion of the proton gradient established by the other four complexes. Protons flow into subunit A, and they then flow into a half channel in the C ring, and this causes the C ring to rotate until the protons meet a second half channel that allows them to exit onto the matrix side of the inner mitochondrial membrane. Since the gamma, delta and epsilon subunits are bound to the C ring, the rotation of the C ring also causes rotation of the gamma subunit, which is actually located or protrudes up into the alpha beta ring. It's this mechanical, physical turning that provides the energy that is required to cause the transitional changes in the beta subunits. As I mentioned before, there are three possible configurations that the beta subunit can take. In the loose configuration, ADP and a phosphate is bound. Then in the transition between the loose and the tight configurations, the phosphate and the ADP react to form ATP. Finally, the tight configuration undergoes a conformational change that causes it to enter the open configuration and thus release the newly formed ATP. Because there are three beta subunits, then this is actually occurring 3 times simultaneously, and the complex sort of cycles between each state, as you can see here. The electron transport systems found in plants and also in photosynthetic bacteria are remarkably similar to those found in mitochondria, but they mainly differ in the fact that whereas in the mitochondrial ETC the source of reducing power comes from products of the citric acid cycle, in chloroplasts and in bacteria this power comes by excitation of electrons by photons of light. The main focus of this tutorial is on the mitochondrial electron transport chain, so we will only look at the electron transport chain in chloroplasts and bacteria briefly. In plants, the electron transport chain occurs as part of the light-dependent reaction in photosynthesis. It begins when a special pair of chlorophyll molecules termed P680 in photosystem 2 are struck by photons of light and become excited. They are passed via a pheophytin molecule to a bound quinone termed QA, and then to a transferable quinone termed QB. This quinone is reduced to quinol and then moves from the photosystem into the membrane. Ferredoxin transports the electrons to the final step of the chain, which is the enzyme ferredoxin NADP reductase. This oxidizes the ferredoxin and uses the electrons to reduce NADP to NADPH using protons from the matrix. Protons are primarily transported across the membrane by cytochrome B6F, and some are released during the photolysis of water by photosystem 2, which is also how P680 gets more electrons once it has been oxidized during photosynthesis. The protons then move back across the membrane via ATP synthase in the same way as oxidative phosphorylation in mitochondria occurs and generates ATP. Photosynthesis in purple photosynthetic bacteria occurs in a very similar way that it does to plants. The difference being that they only have one photosynthetic reaction center, and that the special pair of bacteria chlorophylls in this case are P870 or P960 depending on the species. Photons of light excite this special pair, which transfer electrons via a bacterial phaeophytin through a bound quinone molecule to a transferable quinone molecule that then enters a Q cycle with cytochrome BC1. Electrons are transferred via an iron sulfur protein through cytochrome C1 to a mobile cytochrome C2. The difference is that cytochrome C2 then moves back to the photosynthetic reaction center, sometimes via 4 bound heme C-type cytochromes depending on the species, and transfers those electrons back to the special pair in that reaction center, whereas in plants it goes on to photosystem 1 and the production of NADPH. So there are forms of regulation in the photosynthetic electron transport chain that you don't get in the mitochondrial ETC. For example, regulation by light harvesting complexes. LHC2 is normally found bound to photosystem 2, but when the quinone pool is reduced, it's phosphorylated by the kinase STT7 or STN7 and transfers to photosystem 1. However, when the quinone pool is oxidized by overactivity of photosystem 1, LHC2 is dephosphorylated by the phosphatase PPHI or TAP38 and then reassociates with the photosystem 2. There is also a large amount of regulation using calcium signaling during the photosynthetic electron transport chain. For example, calcium is found bound within the manganese cluster of the oxygen evolving complex of photosystem 2. It's also found regulating the PBSO subunit of photosystem 2 and several other steps, as you can see in the image. I've put a link to the paper that I got this from underneath, so if you're interested in that, you can go and have a look. Now, speaking of regulation, how is the mitochondrial electron transport chain regulated? Well, the mitochondrial electron transport chain is highly dependent on the concentration of substrates, product, and cofactors. You need substrate for it to work, obviously. You need the correct cofactors to be present to allow for electron transfer, and there's also a degree of end product inhibition that occurs within the chain. In a resting cell, there is a high concentration of protons in the intermembrane space, there is a high concentration of ATP in the cytosol, and there is a high concentration of ADP in the matrix. However, as the cell respires, this ATP will be broken down to ADP. This means that there will be an increase in the concentration of ADP in the cytosol, and in turn ADP in the mitochondrial matrix will be used for ATP synthesis. This increases the concentration of ATP in the matrix. This will therefore inhibit the electron transport chain because ATP produces inhibition effects on complexes within the chain, such as complex 4. Also, as ADP is converted to ATP and is transported out of the mitochondria, then there will be less ADP in the mitochondria to allow for synthesis of ATP. As a result of this, a way is needed to maintain the concentrations of ATP and ADP in the mitochondria and the cytosol. This is achieved by ATP-ADP translocase, a transporter protein that exchanges ATP and ADP across the mitochondrial membrane. Here is the mechanism for ATP-ADP translocase. As you can see, alternatively binding one ADP, transporting it into the matrix, then one ATP, transporting it into the cytoplasm. But beyond the concentration of substrate and product, then you need electrons for the chain to function, and these are supplied by NADH. So you need to maintain the concentration of NADH in the mitochondria. Under high activity levels, for example, the NADH produced by the citric acid cycle is not always enough to meet demand for electrons for the chain. This is where NADH shuttles come in. NADH can't be directly transported into the mitochondria, so instead NADH has to be transferred into the mitochondria by a variety of shuttles, one of them being the malate aspartate shuttle. As you can see, NADH is converted to NAD via the reduction of oxaloacetate to malate, which is then in turn transported across into the mitochondrial matrix. The malate is then converted back into oxaloacetate, converting a mitochondrial NAD into NADH, with the oxaloacetate then being converted to aspartate using glutamate. This aspartate is then transported back across the mitochondrial membrane into the cytoplasm and is converted back into oxaloacetate to begin the cycle again. Another way in which you can exchange hydrogen ions across the membrane of the mitochondria is the NADP transhydrogenase. These are independent of the other two regulatory pathways. The enzyme catalyzes the reversible reaction of NADH to NAD and the reduction of NADP to NADPH. In the process, you can see that a hydrogen ion from the matrix is transported across into the cytoplasm or vice versa. There are also a number of inhibitors that can affect the electron transport chain. These can be naturally occurring or synthesized, and they normally inhibit specific steps or complexes within the chain. They've been used to research the roles of each complex and the interactions between complexes within the electron transport chain. So here is a diagram of the most important inhibitors of the electron transport chain. Now I'm going to go through a list of the most important ones. The first being rotenone, an inhibitor of complex 1 that is commonly used as a pesticide. Intoxications by this are very rare, mainly because complexes 2 to 4 are still active, so the electron transport chain will continue to function, but it has to use FADH2 and can't use NADH, and thus the proton gradient will be reduced. So these are some other inhibitors of the ETC that inhibit complex 3. Not really going to focus on these, just going to move on. Some of the big inhibitors of the electron transport chain, and both naturally occurring, are cyanide and carbon monoxide. They both inhibit complex 4, and actually they both affect the binucleate center, but cyanide ions bind the copper, and carbon monoxide ions bind the iron in the heme group found in the binucleate center. Again, oligomycin, gonna skip over this. The only thing to note is that because it inhibits ATP synthase, the last step of the electron transport chain, it will actually inhibit the entire chain, unlike most of the other inhibitors you see. Reactive oxygen species are produced when the chain is inhibited. So oxygen is the terminal electron acceptor, and when only 1 or 2 electrons are transferred to it instead of the usual 4, it produces these superoxide and peroxide radicals that are highly reactive. This occurs mainly at complexes 1 and 3 in the chain, especially complex 3, and are used as a way of showing that the chain has been inhibited is by levels of these. Another thing that can occur to the electron transport chain is uncoupling. This occurs during thermogenesis. There are specific proteins called uncoupling proteins, such as thermogenin, that cause this. There's no ATP produced during this process. the H plus gradient is simply dissipated and lost as heat due to an increased permeability of the membrane. This can be induced by drugs. In fact, dinitrophenol is a banned weight loss drug that acts exactly through this mechanism. It's an ionophore and causes protons to leak across the mitochondrial membranes. This does make it effective at losing fat, but unfortunately it gives it these really severe side effects: sweating, hypothermia, tachycardia, and then eventually complete cessation of heart function. Uncoupling proteins are thought to be activated and therefore have a mechanism that requires long-chain fatty acids to be present. It's thought that the long-chain fatty acids bind into a pocket within the uncoupling proteins, and the negatively charged carboxyl head will bind the hydrogen ion. This binding will cause a conformational change that induces a symport-like transport of the long-chain fatty acid and the hydrogen ion across through the membrane. The hydrogen ion will then unbind from the long-chain fatty acid and be released into the matrix, but because the long-chain fatty acid is anchored into the pocket of the protein, it can't leave, it can't go out into the matrix, so it simply reverts back up into the protein to the position it was in before and can bind another hydrogen ion. The pKa values of the long-chain fatty acids used in this process is therefore key, because if you have a fatty acid that has a lower pKa, this means that it has a higher Ka, i.e. that it dissociates from protons more readily, and therefore you need a higher concentration of protons available before transport can occur. So this is just a summary list of the main inhibitors, including uncouplers of the electron transport chain. However, the electron transport chain we've focused on so far is true for mitochondrial electron transport in mammalian species, but it's a bit simplistic in terms of all eukaryotes. There are some species of protists, fungi, plants, and even some animal species that have other complexes involved in their electron transport chains that give a branched electron transport chain. An example of this is alternative oxidases, enzymes found on the inner membrane of the mitochondria that act both to maintain the quinone pool by oxidizing ubiquinol to ubiquinone, and they also provide a secondary site of terminal reduction of oxygen to water. This means that they provide resistance to cyanide poisoning, but because they act between complexes 1 and 2, complexes 3 and 4 are still inhibited. So therefore, only complex 1 would remain functional in the translocation of protons, and so the proton gradient would be reduced. Alternative oxidases themselves can be inhibited, for example by salicyl hydroxamic acid, or SHAM, and this is being used as the basis for various antibiotics against a type of bacteria called Trypanosoma brucei, which is the cause of sleeping sickness. This particular bacteria uses alternative oxidases as the only source of terminal oxidation in its electron transport chain. There's no complex 4, so this is highly effective against these bacteria. Another class of additional proteins that can be found in electron transport chains are rotenone-insensitive NADH dehydrogenases. They are found in certain species of prokaryotes and even some eukaryote species, including yeast, fungi, and plant species such as species of potato and pea plants. These complexes occur on both the external and internal surfaces of the inner mitochondrial membrane. They are named NDX and NDIN respectively. Yeast and certain species of Ascomycota have equivalents of this, the NDE proteins, which act in exactly the same way. Rotenone-sensitive NADH and NADPH dehydrogenases provide an alternative pathway to complex 1. Their activities are dependent on calcium signaling, with the exception of ND in NADH dehydrogenase. But they don't transport protons like complex 1, so if it was inhibited, say, by rotenone, then there would be some loss of proton gradient and therefore loss of ATP production. This is a diagram showing an overall electron transport chain that you might find in, say, a species of plant that has both alternative oxidases and NADH dehydrogenases. There are a large number of proteins, as you can see, within the electron transport chain, and the genes encoding each protein are subject to regulation by expression. An example of this is hypoxia inducible factor 1 alpha. So normally this is degraded by an oxygen dependent proteasome, but under hypoxic conditions levels of oxygen are obviously too low to allow for this to occur, so levels of this HIF1 alpha protein build up, and it's a transcription factor, so it causes an increased transcription of certain subunits of cytochrome C oxidase, or complex 4. One of these is the CCO7A1 subunit, which is involved in the catalytic activity of cytochrome C oxidase. And along with this, during hypoxia, there is also an upregulation of expression of CCO4I1 and 4I2 subunits. CCO4I1 in particular is important because phosphorylation of this subunit is what prevents end product inhibition by ATP of complex 4. So, as you can see, upregulation of these two subunits is geared at increasing the efficiency of complex 4 in the electron transport chain under hypoxic conditions to try and keep it running. The importance of genetic control can be seen in diseases associated with the electron transport chain. For example, Leber's hereditary optic neuropathy. This is a disease that causes blindness suddenly during middle age and is caused by a series of mutations within the mitochondrial DNA that encodes subunits of complex 1. The actual subunits affected are the NADH dehydrogenase subunits 1, 4 and 6, and this causes improper function of complex 1 and thus causes the retinal ganglia, which are particularly high in energy demand, to begin to degrade and thus leads to blindness. So this is just a brief summary of the points made in this tutorial. It will just flick through, and below in the description I will put links to additional reading and also a link to a Google document which will have the entire script and the referencing for this presentation. So yeah, thanks for watching.